Our cells are constantly working to keep us healthy. They grow, divide, and repair themselves with remarkable precision. But sometimes, mistakes happen in this intricate process. That's where P53 steps in. It's a protein, a tiny molecular machine, often called the guardian of the genome. This guardian has a crucial job, protecting our cells from becoming cancerous. Think of P53 as a vigilant security guard inside each cell's control room. When everything is running smoothly, P53 keeps a low profile. But if it detects any damage to the cell's DNA, the blueprint of life, it leaps into action. P53 can halt the cell's cycle, giving it time to repair the damaged DNA. This prevents errors from being passed on when cells divide. In some cases, if the damage is too severe, P53 can even trigger a process called apoptosis, essentially instructing the damaged cell to self-destruct. This might sound drastic, but it's a vital safety mechanism. By eliminating potentially dangerous cells, P53 helps prevent the uncontrolled growth that characterizes cancer. For a long time, scientists focused on a specific type of RNA messenger RNA, or mRNA. mRNA carries genetic information from DNA to the ribosomes, the protein factories of the cell. This information dictates the order of amino acids, the building blocks of proteins. However, scientists discovered that mRNA is just one piece of the RNA puzzle. A vast majority of RNA molecules don't code for proteins at all. These are the non-coding RNAs, or ncRNAs. Instead of carrying blueprints for proteins, ncRNAs play diverse regulatory roles in the cell. Imagine them as skilled conductors, subtly adjusting the volume and tempo of various cellular processes. Non-coding RNAs, despite their name, are far from silent partners in the cell. They are deeply involved in controlling P53's activity. These ncRNAs can interact with P53 in various ways, fine-tuning its response to cellular stress. Some ncRNAs act like boosters, enhancing P53's ability to bind to DNA and activate its target genes. This increased activity leads to a stronger response against cellular damage. Conversely, other ncRNAs act as brakes, preventing P53 from being overly active when not necessary. This regulation is crucial to avoid unnecessary cell death or premature aging. Section 4. A complex interplay. Examples of non-coding RNAs and P53. The world of non-coding RNAs is incredibly diverse, with various types playing unique roles in regulating P53. Let's meet some of these key players. MicroRNAs, or miRNAs, are these tiny RNAs that act like molecular switches, turning off the production of specific proteins. Some miRNAs can directly target P53's mRNA, reducing its production and therefore its activity. Long non-coding RNAs, or lncRNAs, are longer RNA molecules that can act as scaffolds, bringing together different proteins, including P53, to specific locations within the cell. This organized assembly enhances P53's efficiency in responding to DNA damage. Pseudogenes, once considered fossil genes with no function, are now known to produce non-coding RNAs that can directly interact with P53. These interactions can either enhance or inhibit P53's activity, showcasing the complexity of this regulatory network. Section 5. Guardians of Health, Non-Coding, RNAs, P53, and Disease Prevention. The intricate dance between non-coding RNAs and P53 is vital for maintaining cellular health. When this delicate balance is disrupted, it can pave the way for diseases, particularly cancer. In many cancer cells, P53 is mutated or silenced, preventing it from carrying out its protective duties. This allows damaged cells to proliferate unchecked, leading to tumor formation.